Okay, good morning, everyone. I'm hoping that you can see and hear me now. We fixed that lightning quick. We had a little blip back there, but hopefully it's all great now and you can see us. And I want to say a huge welcome to our annual Drug Repurposing for Rare Diseases conference. And we're really excited to welcome you online this year. Usually we'd be doing this around February, but today we're online and we're doing it in the summer and we're raring to go. And we've got 2 days full of talks and discussions from across the rare disease community, highlighting a range of repurposing projects and experiences. I'd like to say a big thank you to our sponsors this year. So we've got our headline sponsor of LifeArc. Our corporate partner this year is Helix. Our gold sponsor is the Evolution Education Trust. Our silver sponsor, Pulse InfoFrame. Another silver sponsor, Purposeful. And our bronze sponsor, Pharmacomedics. Thank you so much for sponsoring us and helping us to make this event a reality. So why are we here? Everyone hopefully knows a little bit about rare diseases today, but obviously in the UK, approximately 3.5 million people live with a rare disease, which can be chronic, life-threatening and isolating. And a rare disease is defined by the EU as affecting fewer than 1 in 2,000 people. And we're here today because there's a real need in the rare disease community to find treatments and health technologies for those living with rare diseases. Of the 7,000 recognised rare diseases, only 400 have licensed treatment. And at the current rate of drug development, it would take 500 years to get treatment for all these conditions. We really want to see this sped up for the people out there that need treatments. And that's why we're here today, to bring people together and talk about how we can do that as a community. Now, who are we? If you're new to Find a Cure, then hello, we're really glad to have you here. If not, then you'll have heard me say this before or someone else say this before. But we're a UK charity that is building the rare disease community to drive research and develop treatments. Our vision is a world in which all rare diseases have treatments made together with patients for patients. And we really put the patient and the patient organisations and advocates at the heart of everything that we do. And how do we make this happen? Well, we make this happen through a very dedicated and wonderful team, if I say so myself. And you'll be seeing a lot of these people on the screen over the next 2 days. So we've got Rick, who is our resident drug repurposing enthusiast. He's our CEO, and he'll be talking to you a lot over the next 2 days, taking us back to some of the basics of drug repurposing. You've got me, I'm the projects coordinator. We're joined by Blaine, who is our marketing and engagement manager. Mary Rose, who is the head of operations and has been working very hard behind the scenes to make this a reality. Laura Thompson Harper, who's our fundraising manager, and Hannah Harvey, our newest recruit, who is the projects administrator. And how do we put patients at the heart of what we do? So a lot of the work that we do is our patient engagement, a patient empowerment program, which is based off patient group training. We try and make people develop in these 4 areas if that's what they want to do. So we help people to build and grow their organizations, raise awareness for their rare conditions, get ready for research, and support their patients and families. And we do this through a combined training program of workshops, webinars, our patient group mentoring program, and our e-learning resources. We also like to think that we help to unite the different people in the rare disease space, and we do this because we think that collaboration really drives the rare disease community forward, and no one can do anything alone. So we do this through our Rare Disease Showcase series, which we also held online last autumn-winter, our Rare Chat series, and the Student Voice Prize essay competition, which brings medical students together with rare disease advocates. And of course, our drug repurposing work sits alongside everything else that we do. So our annual conference highlights the best examples of drug repurposing in the field of rare diseases. And we think it's equally important that we create opportunities for you to meet new people outside of maybe your field, outside of patient groups, opportunities to share your work and to get your voice heard, and opportunities to strengthen the rare disease community and to actually make collaborations that you might not have expected, but can actually help you achieve what you want to. And we do that by having a really diverse set of attendees to these events. So we've got people from pharma, we've got patient groups, we've got consultancies, CROs, biotechs, clinicians, academia, you name it. We like to make sure everyone feels welcome and can join the discussion and can join the conversation. So in terms of our drug repurposing, it all started back in 2014 at the inaugural scientific conference and was actually one of the first events organised by Find a Cure. We like to think that the event has really grown since then, away from just being scientific to being something that everyone can get involved with, where there's something for everyone and everyone can relate to and learn from. We've now had 7 years of the conference, 6 venues and hundreds of attendees. Last year, it was actually our last in-person event, and we welcomed over 125 attendees to the American Square Conference Centre in London. And I think this quote really sums up why we love this event, in that it brings together all those different viewpoints so you can see what it's like to live with a rare disease, as long as getting the the viewpoint from pharma, from industry, from academia. And that's something we really want to build and grow this year. We really want you to get involved with having those discussions. So if you were here for our virtual Rare Disease Showcase last year, you might be familiar with our Swapcard platform, but I thought I'd just show you how to make the most out of it as well. So to watch the live sessions, which you're watching now, you can go onto this live session watch now button, and it will follow you around the site, so you can go and look at other things and you'll still be able to see it in the corner. We'd love for you to get involved by submitting questions to our speakers, having chats with other attendees, and also answering polls so we can find out a little bit more about you. You can look at the agenda for both days in the agenda tab. You can just select your day up at the top here, and also add your favorite sessions to your schedule, which you can look at in the My Events bit. It really makes sense to register events so you don't miss out on them, and to also kind of get sorted, get your head straight with where you're going to be at what time. Breakout sessions will all be hosted in Zoom, and they're designed to be interactive, so it'd be great if you could get your cameras on. You will need to register for these first, and you can join from the breakout page itself, or there'll be a link in your notifications as well. You'll be able to ask questions directly to the speakers in this session, so you can unmute yourselves and do that. I've just shown you this little screenshot here, so you can clearly see how to join there at the bottom. This year for our Drug Repurposing Conference, we've got the Patient Group Poster Zone, where we've got some really fantastic examples of patient-made posters highlighting different types of rare diseases, different types of treatment options, and it'd be great if you could check those out, because there's been a lot of hard work put into those. And also we've got our exhibitor resources, where you can see examples from our different exhibitors, examples from Find a Cure, videos, and plenty of resources to keep you busy in the interim between sessions. Obviously, we want you to connect with everyone, and we think Swapcard's a really good platform to allow you to do this. So you can check out other attendees and find and connect with people, download their details after the event, so you can keep them stored there. You can have text chats with people or organize video meetings in this little text blurb section at the top of your Swapcard. You can go to the exhibitors tab and talk to exhibitors within there. Post on discussion boards, get involved in the chat, ask questions. And also we have a lovely recording from our virtual Rare Disease Showcase in the exhibitors resources, which is how to make the most of digital networking. And you can check that out on our YouTube as well if you ever want to reference that in the future. If in doubt, you can find our platform guidance on the homepage, which should run you through everything you need to know about how Swapcard is going to work for this event. Just a little bit of housekeeping for the Zoom sessions. They are all being recorded, but we're only going to record speaker content, so no attendee questions or breakout discussions will be included, so you don't need to worry about that. Make sure to have your cameras on in the session, but please mute yourself when you're not speaking so we don't have any background interference. If you do have any technical difficulties, the Find a Cure team is always on hand to help, so you can just put that in the chat and we'll get it sorted as soon as we can. And then you can raise your hand at the bottom as well to show the host that you want to ask a question or speak. Now we are trialing out some subtitles in the Zoom breakouts this year. They're not perfect, but they're not too bad. So hopefully they'll be of use to some of you. If not, you can turn them off, and I've just shown you here where you can turn them off in the live transcript bit at the bottom bar. And also you can view the live transcript if you want to kind of read through and just catch up on what somebody said. So hopefully they'll be of use to some people, and you can always turn them off if you don't fancy. Anyway, after that long introduction, and hopefully you remember all of that, I would love to welcome you all to the main event, and I really hope you enjoy the next 2 days of what we've got planned for you. I'd love to now introduce Rick, who's going to take us back to the basics of drug repurposing and tell us why we should care about drug repurposing. So thank you, everyone, and enjoy the conference.
Thank you, Phil. Really excited to be here, everybody, and glad we got through that first little glitch at the start. Phil's kind of set the scene really beautifully. You've got a good sense of why we're here at this event, and my aim today is hopefully to convince you should care about repurposing. If you're here just because you've heard about it and it sounds intriguing, the next hour and 45 minutes is going to be about convincing you this is a process that matters for rare diseases, and something we're going to dive into in much more depth over the next day or two. So I'm going to try and share my slides, and we'll dive into the excitingly titled session, Back to Basics. What is repurposing and why should we care? So hopefully you can see my screen, and you can now see my screen in full presenter view. I'm getting some nods, which is great. So this session is something I've been thinking about for a little while. And the aim here is to take you on a delightful guided tour with me as your guide and some fantastic clinicians and academics to just dive into the content of repurposing and help you understand the very basic things and also the reasoning, as I said, why we're here. So I'm Rick. Most of you will have heard me talk far too much already, and you're going to hear a lot more of it over the next few days. But I am CEO of Find a Cure. I've been in charge for, I think, around 4 years and at the charity for about 6 years, which is a little scary at this point. I'm also the non-branded member of the team today. Everyone else has got a Find a Cure banner. I don't get one. I get to show you my Liverpool Football Club allegiance, so enjoy that. I'm really happy to be here, and I've done a number of these talks now, and this is a topic that really matters to me, and I think a topic that's increasingly becoming important in the world of rare diseases. At this stage, it'd be good to introduce some of our other panelists. So Rona, if you'd like to unmute yourself and say hi, we'd appreciate it.
So good morning, I'm Rona Smith. I'm a Senior Research Associate at the University of Cambridge, and I'm also a consultant in nephrology and vasculitis. Much of my work pre-COVID was working with patients with rare disease, a form of vasculitis, and I've now ventured into the COVID world, and I'm currently Chief Investigator of one of the urgent public health prioritised national trials called PROTECT-RE.
Thank you, Rona. That's fantastic. And we're diving into that work on COVID and rare diseases in more detail quite soon. John, would you like to say hi?
Hey, I'm John Liddycote. I'm a Senior Research Associate as well, but I'm in the Faculty of Law. I specialize in drug repurposing and intellectual property, so patents and things like that. But I won't drone on too much about the law today. Instead, I'll talk about some empirical data, which is hopefully a little more entertaining.
Thanks, John. I'm excited to hear what you've got for us. And I'm sure everyone would love to hear about the law as well, but don't go too far quite yet. And thank you. And then Indy as well, if you could say hi.
Oh, hi. I'm Indy Banerjee. I'm a consultant pediatric endocrinologist. I work at the Children's Hospital in Manchester. My rare disease interest is the condition of hypoglycemia called congenital hyperinsulinism. And that's a big problem for us, but we've been involved in having drug trials and different types of studies in this research area for a long time. And I'll talk to you about my lessons that I've learned along the way and how that applies to repurposing. So really looking forward to the rest of the conference.
Thank you so much, Indy. That's brilliant. So as you can see, we've got a nice panel assembled, and the intent today is to take you through a winding narrative, moving through each of the different stories from our speakers here to give you a sense of why repurposing matters in this context in rare diseases and why it's actually currently a really big topic. Please feel free to ask questions. Drop them into the chat within swap cards, and they'll be fed through to our panel here. Things that are popping up during the narrative, we may drop in major questions, particularly things to clarify points. The hope is to have about half an hour towards the end of this session, which will be open discussion where you can ask whatever your questions are to this panel, and we'll get some really good discussion going between you, our audience, and the guys here. But it falls to me to introduce this topic and dive you through some of the very basics, and hopefully to convince you that you should care. Why should you bother to repurpose drugs? So to do that effectively, we have to use my favorite classic slide that I've shown hundreds of times before to make sure everyone's on that same page. What is drug repurposing? Fundamentally, at its most basic level, repurposing is just recycling. And we all know recycling is a good thing. It's finding new ways to use existing medicines to increase the efficiency of how we're using our existing complement of drugs that are available to us. It's taking a drug that's approved for one group of patients, ideally generating some really good clinical evidence to show it's effective or efficacious in a completely different patient population. It's very simple. It's a concept that goes by a whole range of different names, whether reprofiling, repositioning, some people might refer to it as license extensions, but fundamentally, whatever you're talking about, this basic premise of recycling underpins what we're doing in the repurposing field. So what does that kind of mean? What's the implications of this? What's the ethos behind repurposing? And the first thing is that word I've mentioned already, efficiency. It's about more efficiently using the resources that are available to us as a society, as a planet. But it's also about trying to drive essentially the efficiency in how we deliver drugs to the patients. It's finding efficiencies in the research process and the development process that brings these treatments to the patient themselves. And that can only be a good thing, because efficiency generally means you're increasing the speed or reducing costs. And that's something we're all about. Another key theme when we're talking about repurposing is going to be availability. If you're repurposing, you're using a drug that's currently out there. There's something available that you can use where there wouldn't be if you're creating something completely new. Generally, though, that drug's likely to be more widely available to people to research on it, and in many cases more widely available to the patients themselves. It's probably may already be available on the market, and in most cases that's the case. And it also might be the case that this drug is more widely available in a global sense, so to a whole range of different countries, or might be present in one of the major drugs that's available in countries all around the world, Africa, Antarctica, wherever it is. It might be that basic drug that's in the kit that you need to keep people moving, and that can be a really good thing to ensure we have patient access. Repurposing is also about knowledge. It's about making the use of the knowledge that we already have, finding out how we can apply that information to speed that process and drive efficiency. It's about using what's there and generating new knowledge to provide the evidence that we need to effectively use these drugs in different indications. And I think this is actually a process of innovation. Lots of people wouldn't think of repurposing when you talk about the word innovation. People think of something new, and they think that that doesn't involve those new uses, but actually innovatively using what's out there is a really crucial way of doing things. And these are a great level of innovation, how we think about things and also how we deliver trials, because ultimately the aim is to get a drug to patient, and repurposing can help you do that more quickly. And finally, and crucially, it is actually still about evidence, generating as much high quality, crucial evidence of its impact as possible, really making sure that although we're trying to deliver more efficient trials, we're still having a high bar of evidence that's been generated and a high threshold to make sure we deliver real benefit to the patients themselves. And evidence should always be at the forefront of our minds in any clinical development pathway. So if you're thinking about repurposing, we have to think about what drugs you can use. And I suppose a simple answer is anything that's out there. But we can look at it in more subtleties and look at some of the different classes and think about what might be available to us that wouldn't otherwise have thought of. An obvious pool of pharmaceuticals for repurposing are generic drugs, drugs that are currently out there on the market that are not currently protected by intellectual property and are therefore quite readily available and produced by multiple companies. And these are great fodder, if you like, for the repurposing process. Alongside that, you'll have currently branded compounds. These are compounds that still retain intellectual property and so are produced by a single company. And the third kind of pool that we can tap into for repurposing are shelved compounds. So these are compounds that are currently present in a pharmaceutical library, which might be a fridge or a shelf, who knows what that looks like. But essentially, it's been through the trial process, generally will have been proved to be safe for human use, but it didn't prove efficacious for its original targeted illness. So it's not currently made it to the market. It's sitting there as a resource that's untapped, and we need to find ideally uses for this separate from its original indication that can bring it to market and make a difference to more patients. And these are all great sources of repurposed drugs that we could dive into. So why does the source matter? Why am I talking about this? Firstly, it's crucial to know that there are different sources that you can look at, and many people may not consider, for example, shelf compounds as something to look at for your innovation process or thinking about finding a candidate for your condition. But secondly, and more importantly, the type of drug that you start with will affect how you're going about the development process, how you can move that research forward, and ultimately how it reaches the patient. It affects your freedom to operate, whether you can actually access the drug for research or not. It will affect the route to market, how you can think about securing maybe intellectual property to get a patent, and how that makes a business model for you to reach those patients, and which other companies might need to be involved. And all of these considerations will feed in ultimately to the cost of the drug to deliver it to the health system. And dependent on what angle you're coming from as a researcher, that might be something you need to consider really carefully to ensure you can get that patient access. So let's look at these three classes in a bit more detail and think about how the drug that you start with might affect where you go moving forward in your repurposing project. So let's start in the middle. Current branded compounds. So in these cases, we know that there's going to be some intellectual property intact. And what that means is someone has a patent on it that says, I had the idea to use this drug or to create this compound and use it in people in this way. And that means there's a parent company that are owning it, and they're the only people that can make it at this stage. So this gives a really clear business model to move this type of repurposing project forward. They are simply trying to get a new license to make this drug reach more patients, and that plausibly is a really potentially beneficial thing. Other companies therefore can't really work with these drugs very easily, and you certainly need permission or collaboration with that parent company to move it forward if you're looking at academic research into these drugs. What it also means is when it reaches market, if it is repurposed, that final price is likely to be very similar, if not exactly the same, to the current licensed form. So it gives some predictability in that, though that may or may not be something that works for your indication depending on its current price. This type of repurposing is obviously great for industry, and there is a clear business model for it, as we said. But sometimes it's seen as risky, and a company might decide it's not worth the risk to do more tests, more clinical trials in a different patient population that might lead to more adverse events, which might lead concerns in their main business, or could just be something that's not really worth that additional risky investment, something that may not be developed effectively. So this may not always happen, and often it can be somewhat slow to get things moving. The next category is shelved compounds, things that are on the pharmaceutical shelves and not currently on the market. And here you can make use of that existing research data from that failed research program. So it gives you a really great way in to start thinking about the drug repurposing project. You may well still have intellectual property, which would give you a really good, strong business model, but also that might have lapsed, and that might mean moving these compounds into the rare disease space, the orphan space, could be attractive, as the companies may then be able to access an orphan drug designation, which gives a series of benefits that can make the development of rare disease compounds more appealing. And in particular, it can give exclusivity on the data from the research. And that's a key thing for many people when they're looking at the business model to repurpose drugs. What we have seen in this space, which is really great, is that big companies are generally opening up these libraries to collaboration, and they're actively seeking collaborators either with small companies or with academics to try and move that early stage development process forward, which makes it a really interesting and lively environment for research. But ultimately, the compound will reach the market as a completely new product, and that makes it, more secure and positive for the companies themselves. It's a collaborative form of research, and that means a clear business model. So that's a great thing. But because you have that collaboration, there are benefits to the partners who are collaborating on it, or you may have some say in the route to market or the final price, which is interesting. And there's less risk to those companies compared to the other thing we've talked about, because it's about recouping that investment they've already put into the drug to get it where it got in the failed trial. And this is a really nice opportunity for them. And I'm really pleased to say we're going to have a talk on the main program tomorrow from AstraZeneca talking about their system, their open innovation system, to use this type of repurposing of their shelf compounds. So do check that out. So the final class of drugs, an area that I think is really important and great for repurposing, and that's generic drugs. In generics, that the intellectual property on the compound itself has expired. You don't have a patent to say, I created this compound. All you can plausibly do is get a patent to say, I can use this drug in this way or for this condition. And I'm sure John will dive into that in much more detail in his talk in a little while, and we'll talk about the pros and cons around that. Suffice to say, that isn't such a strong patent and makes a weaker business model. So we generally see companies are much less likely to work on generic drug repurposing. What does happen with generic drug repurposing, though, is the drug is generally widely available. You can access it, and this means it's available for researchers, it's available to academics, it's available to research driven from patient organizations. So it's great for that type of model of research. And generally, again, because the intellectual property is lapsed, anyone can produce the drug, and this means there's competition, and this will drive down drug price. So generally, generics are going to be lower cost, and that's easier for a healthcare system to afford. You also have often extensive history of human use in your generic drugs, and that means you have way more knowledge than you generally have on another compound. You'll know about the impacts of this drug in different patient populations and a whole range of different side effects that are present for it. And this, again, gives you a wealth of knowledge for repurposing. It's definitely on the rise as a form of repurposing, which is great, and it's prevalent in the rare disease field. And I think we're going to talk about it a fair bit over the next couple of days. And that's really driven by the high unmet need from rare disease patients. But it's fair to say there's no easy route to market for generically repurposed drugs, and that's because of this lack of intellectual property and a lack of financial incentives for industry in particular to work in this area, and maybe an uncertainty within the academic community of how you move from doing your basic research to actually deliver a drug to market. And that's something we'd really like to help to change, and something we're hopefully going to discuss a huge amount over the next couple of days. When you're looking at generics, there is a route that industry can pursue, or you might need to pursue in your work, and that's reformulation. What reformulation essentially means is making a change to the compound itself or the way it's delivered so that you gain intellectual property. That might be changing the chemical structure or the companion compounds that go in the tablet. That might be about just changing the dose and increasing it significantly. Or it could also be changing the way that drug is delivered. You might shift something from a pill, for example, to an inhaler, which might be really crucial for the rare disease that you're working on. And in all of these cases, you're gaining intellectual property, which gives you a more clear business model to move things forward. But by doing that, you require new data, particularly on the safety of that reformulated compound. And this means you're cutting out some of those efficiencies we've talked about. You don't have data there, so you lose access to that past information. So this can slow down the process and make it more expensive. The other thing to talk about and touch on in the generic drug space is the fact that if you can't have a clear business model for a compound, what you might end up looking at is an off-label route to the patients. Now, off-label prescription essentially is all about the idea of prescribing a drug for a condition that it doesn't have a license for. This means it isn't regulated or legally approved to be used in that condition. And essentially, this is something that's done at the clinician's own risk. It's actually a really common practice, particularly in certain fields, for example, in pediatrics, because many drugs are just never trialed effectively in a pediatric child population. Then issues with off-label is rules around it can vary from country to country. And because of that lack of regulation and built-in monitoring of drug safety, there's a lot of uncertainties around off-label use, and it can lead to patchy access for patients. If this is a situation your rare condition is in or something you're targeting, you have to really think about that very carefully and how you're going to address that effectively to ensure patients can access a treatment that matters to them. And again, I think this is something that Indy and possibly Rona may touch on later in their conversations. So that's some of the basics and background around the core concepts in repurposing you need to be aware of. So the next question is, how do I get an idea? All right, it sounds great. Yeah, let's go and do it. But how do we do this? How do we find a drug that might work for my rare disease? There's lots of different approaches to this. I'm just going to touch on 4 major areas that I see repurposing concepts being formed in. And the first and probably most traditional is what I might call clinician or clinic-led innovation. Now, this might be spawned from serendipity, a serendipitous event that by chance someone takes a drug and it happens to make a huge difference to their rare condition when it was thought to just help a simple symptom. And the classic example for this is the repositioning of thalidomide into leprosy. So thalidomide obviously has a really dark history, traditionally prescribed for morning sickness and led to extreme malformations and in the developing embryos of the mothers who were prescribed the drug, and the most profound adverse event, if you like, or serious horrible effect of the compound being used. So it's banned completely. But out in Israel, in a leper colony, a doctor happened to prescribe thalidomide to a patient who just couldn't sleep due to the symptoms of the condition to try and help with this. And by chance, it so happened that this drug quickly helped to show an improvement on the underlying leprosy itself and the symptoms of it. And this led then to further clinical research to move thalidomide into this indication and area and pull this dark history drug into a new area where the risks there can be more effectively managed. So clinic-led innovation might be about a serendipitous events, but more hopefully and more systematically, it should be based upon the knowledge and expertise of the clinicians working closely with patients to understand what options might be available for them and thinking about ways to move things forward to try and give patients a treatment option. Not very systematic, but something we do see a lot of. To be more systematic, we want to maybe look at things like screening. looking at compound libraries, huge collections of different compounds of drugs that we try and compare against a simple model system for our condition. And that model system should recreate part of the underlying biochemistry or symptoms of the condition. And there should be a simple trigger within it or marker within it to show that a drug that you're testing in that system might make a difference. And by having that simple system, you can do a really high throughput, high volume number of compounds and hopefully find one or two that can make a difference to your condition and drive your research forward from there. And tomorrow we'll have a talk from Tim Barrett from the Birmingham Children's Hospital, who used a screening approach to identify sodium valproate as a potential treatment for the rare condition Wolfram syndrome. Another area to talk about is literature mining, which is something that's been growing a lot over the last few years. And this can be as simple as, firing off a Google search to search for papers online that might say something about your condition. But more accurately, it's actually a complex application of artificial intelligence and computer analytics to dive into the meat of the wealth of scientific literature out of there, and to try and find connections between conditions and drugs. And with the technologies that are advancing today, we have AI technologies that can actually infer connections that have never been identified before simply by looking at the raw language that's present in these papers. It's a really exciting area and something we're seeing increasingly developing, and companies that are following this path to identify new opportunities. And this really feeds into that final bucket I want to talk about, which are wider AI approaches, particularly using big data like omic data. So looking at all the genes in the body, the genome, or the proteins in the body, the proteome, or the transcribed genes, transcriptome, etc. And this wealth of information can potentially identify signals that are unique to a rare condition, and potentially signals or changes in the expression that are unique to a drug. And these approaches essentially try to use artificial intelligence to pool all this information together to identify new opportunities in rare conditions or any condition for repurposing. And again, we'll have examples of that throughout the sessions today and tomorrow, including two of our sponsors, Helex, who are running a session later today, and Purposeful, who are talking tomorrow. So do have a look at that and think about this as a way to come up with an idea. Okay, Rick, so you've been banging on for a while. You've hopefully talked about some of the core information that's needed. But what we really are here for is to understand why I should care. Why is repurposing relevant to me? I like my medicines new and innovative. Why should I bother to repurpose drugs? And that's a fair question. Okay, if we're looking out there right now, the things that we're hearing about in science, the things that are exciting, the things that are sexy science, if you like, are shown on this screen. As a scientist, you need to get papers. That's what you're incentivized for. And coming up with a completely new compound, de novo drug discovery, that ticks that box as an exciting, newsworthy event. Finding a new compound to move into people, that's going to excite people. Understanding all of the genes in the body, how they work together, how they turn on to create a human. This is the science that's driving everything forward and is hugely important in the field of rare, where around 80% of the conditions have a genomic basis. It leads us into this concept of personalized medicine, right? Of being able to try and tailor the treatment to the specific mutations or specific condition that a person has, and really have a tailored approach to treatments. And finally, we can see on the horizon this attempt to actually begin to edit and change and modify the genome of patients with genetic conditions to completely rectify or adjust that problem. These are the things that we think of for new and innovative approaches, and that's true. But the first thing to say is that, all of these processes, while exciting and having a really crucial place in the future, are slow and take time and money and may not be able to deliver quickly for patients. And when you're living with an ultra rare condition, what you want to see is something that's going to make a difference to you soon. And you can't afford to wait 10, 20, 30 years for that final gene editing program to come into position. The second thing is to look at, where do these things sit? Where does the industry space sit right now? In terms of drug development. And this is old data that I've talked about many times before, but it really gives a clear view of some of the situation in drug development. In the red bars, we can see essentially the number of new drugs that have been approved year on year, and as I say, this is going back to 96 through to 2013. And there's a rough trend for a decline there, and certainly a stabilization. We aren't seeing a sudden growth and increase in new drugs being created. The green line shows that R&D spend per year in US billions of dollars. And for a period where we're seeing a reduction or stabilization in drugs created, we're seeing an increase in the R&D spend. So this simply tells us it's getting more expensive to create new drugs. And the blue boxes at the bottom here confirm that. We see in 96 a cost of around $0.3 billion to create a new drug. We've got a peak on this data set in 2007 of $2.6 billion. In 2013, $1.8 billion. And the costs now float around the $2 billion mark. These are significant sums to do this type of research. It's not necessarily quick and it's costly, and that isn't necessarily the model that's going to drive the way to speed this 500 years to deliver new treatments for rare conditions. We need something to complement this, to work alongside this. Furthermore, if we look into the pricing in rare conditions, there's a known situation here, right? And this graph just illustrates it simply. Even if we just look at 2016, in the dark bars, we see the price per average patient per year in dollars for a non-orphan drug, a drug for a non-rare condition. And that price sits just below $28,000 per patient per year. For an orphan drug in a rare condition, that cost is over $140,000 per patient per year. And there's many, in many cases, good reasons for that price discrepancy. And we need a price discrepancy in a way to ensure that people are incentivized to deliver drugs for rare diseases, but that isn't something we can sustain across 7,000 different rare conditions, and that's clear. We need something that has a chance to deliver drugs at a slightly lower cost to patients and slightly more quickly to patients. And that is why I believe we should care about repurposing, and it is really relevant to rare diseases. Repurposing is all about working with what you know, okay? It is a process that is fast and cheap, and therefore it should be good for rare conditions. We eliminate the need for that de novo drug discovery process, which is itself clearly highly expensive and time consuming. We're working with a drug that has a known safety profile. We know what the side effects are. If it's generic, we have a wealth of history of human use. All of this gives us information that can help reduce the requirement for early stage clinical trials. Speeds the process, reduces the investment required, makes it quicker, helps you get to patients more quickly. We know about the pathways of action in a repurposed drug. We know the target and how that drug works and delivers. This means if you understand even the basics of your rare condition, you can begin to identify a suite of drugs that might work on this pathway and could have potential for your condition. You reduce the need to screen 10,000 different compounds potentially, and can screen 20 or so that could make an impact. Again, reducing the time and the investment that's needed to get the candidate ready to the clinical stage. And that's really what repurposing is about and why you should care about it in rare. So repurposing, why bother, right? It generally offers a route to deliver medicines more quickly to patients. And that's fundamentally what the aim of drug development is about, to get something in the hands of patients to make a difference to their life, quality of life, or life duration, very simply. It should accelerate the stages of drug development before you reach the clinic. When you get into the clinic, we still need to generate really high bar evidence to demonstrate efficacy, and we shouldn't be cutting corners in clinical trials, but you can get to that stage where you're dosing patients more swiftly with more confidence based on the evidence that's already out there. Because of this lower bar to entry in that preclinical stage, it opens up research to academic-led and patient group-led research. And this in turn can create the opportunity to really target the unmet need in underserved patient communities like those in the rare disease space, where the patient is really engaged in research and really engaged in making sure that their needs are highlighted, and clinicians are increasingly aware of that. And companies too are increasingly aware of that. And all this means there's the potential with repurposing, with the right business model, with the right incentives, to deliver orphan drugs at lower prices, more affordable prices, which could be complementary to the existing model we've got, with some of those absolutely purely exciting innovations we're seeing in genomics, in personalized medicine, in gene editing. So I say, why wouldn't you bother with repurposing? Now that's my pitch to you, and hopefully I've convinced you of the importance of this. And what I'm going to now do is turn to my panel of experts to really dig into this in more detail and look at some examples. And the first place we want to go is what's been happening to us all over the last year and a bit, which is this COVID pandemic. And, you know, this is why I'm working in my office with my Liverpool shirt on the wall rather than in the office with the team right now. We are living in a different space. And what we have seen is repurposing has kind of come a bit to the fore, because it's been a route that's been used to address that COVID pandemic. And I think it's really interesting, and we can learn a huge amount from this emergence as a rare disease community. And there are absolutely parallels, really strong parallels between rare diseases and emerging infections, which I want to draw on, and which I'm then going to pass on to Rona to really dive into in more detail. In rare diseases and emerging infections, we have really high unmet need. We have little knowledge of the disease at that point of emergence. And to be honest, even now, probably our knowledge of COVID is still, you know, very much in development. And we have few interesting clinicians and researchers. That's a common point at that point of emergence. But what we have from the patients who are affected by these conditions is a really strong need for rapid development of a treatment. And if you're going to try and develop that as a researcher, you need to reduce some of those unknowns, because the unknowns about the condition are really pronounced. So using a drug that you know something about is going to help. It's fundamentally obvious. So repurposing drugs in both of these situations can help you deliver for that scenario. And that is one of the reasons why we've seen repurposing take the stage around COVID, and why I think it's crucial for rare diseases. But if that's the case, why haven't we seen, you know, hundreds of different repurposed drugs into ultra rare conditions so far? And unfortunately, there's a key difference here, and that key difference is urgency. We're looking at diseases that are transmitted in very different ways. We have generational transmission, inheritance, essentially, in the majority of rare conditions. And people on the street aren't worried that they are going to be affected in their lifetime by that condition. With an emerging infection like COVID, there's that sense that anyone could get it. It could affect anyone, because you have that societal transmission. And this creates that sense of urgency and drive to make a difference. And what we need to try and do, I believe, as a rare disease community, is generate some more of that urgency, because the urgency felt by those patients is absolutely no less, and probably much more in many cases than those who live with COVID. But we've seen that urgency drive a huge level of investment and a huge level of regulatory flexibility in COVID that we want to see captured more in the rare disease space. So with that, we're going to move from my big thesis on why you should care about repurposing to look at more detail about what's been happening over the last year with Rona and her work repurposing for COVID.
Well, thank you very much. And I hope everybody can see my slides. Great. Okay. I'm getting nods, which is good. So what I'm going to talk about over the next 10 minutes or so is about, sorry, I can't see my own slides. So that's why I'm just being slightly hesitant here. Let me just see my own slides.
You pop into presenter view, Rona, that'll probably make a difference for you.
Hold on 1 second. Apologies. I should be good at Zoom by now, shouldn't I? Anyway, that's fine. I can look at them on my other screen. So do not worry. So I'm going to talk about the next 10 minutes about repurposing in COVID. And what I'm just going to cover are my slides advancing. Apologies. Yes. What I'm going to cover is why drug repurposing has been so important in COVID-19. Think about the core concepts to do with drug repurposing, and then just think beyond COVID-19. What can we learn? So as Rick has already said, the key driver behind drug repurposing in COVID-19 is the urgency of a global pandemic. And the urgency really stems from the fact that to develop a new drug for a condition, then this would normally take between 10 15 years. We just did not have the time for that in COVID-19. And therefore, we have therefore looked to do drug repurposing. So drug repurposing means that drugs could become available within 3 to 5 years. And actually, COVID has accelerated that even further, with drugs becoming available within 6 to 12 months. Really quite an impressive achievement. Now, I hope my slides have moved on and we're looking now at core concepts. If somebody could just signify that that is the case. Yeah, excellent. So I just want to talk about core concepts to do with drug repurposing in COVID, and then I'll draw this out to what we can apply to rare disease. So before we talk about any form of repurposing, we need to think about the scientific rationale. Why are we selecting a particular drug to trial in a condition? And then the next stage is really well-designed clinical trials. And as Rick had already mentioned, many repurposed drugs are used off-label. And one of the drawbacks of using off-label outside a clinical trial setting is really we're not collecting the data being able, and therefore using the information gathered by trialing that drug to inform future decisions. And in order to be able to do well-designed clinical trials, infrastructure and collaboration is absolutely key. So let's just look at the scientific rationale. So in terms of scientific rationale, there's two elements to think about. One is the drug and one is the disease. So in terms of the drug, we know about the target for that drug. We know about the PK, the pharmacokinetics. We know about the safety profile of a drug. In terms of the disease, and particularly in terms of COVID-19, actually, when it first hit, we knew very little about the disease. We have learned enormous amounts over the past 18 months. And actually, we need to know about the disease and the pathogenesis to understand where to place a drug in the disease course. So, for example, for COVID-19, it is a spectrum of disease. There is an early stage where most patients are outpatients at the point of viral infection. And then what became apparent quite quickly with COVID-19 was that there was certainly a later phase, this inflammatory condition. And therefore, drugs that are used in the early phase most probably are not useful in the later phase. And this slide here shows drugs just in the first 9 months of the pandemic that were considered at the various stages in COVID-19. Drugs in red are drugs where there has been no evidence that they offer benefit. Drugs in green, namely corticosteroids, dexamethasone and heparin, have been shown in big trials to have benefit. And those drugs in yellow, such as remdesivir, have a benefit in certain populations. So how do you go about choosing which drug to use? And Rick has already talked about this in detail, and I'm not going to focus on this in my talk. But certainly at the beginning of the COVID-19 pandemic, we knew that this was a coronavirus infection, and therefore very many of the drugs focused on repurposed, approved antiviral drugs, looking at blocking virus entry or protease inhibitors, and then virus release blockers. And later, when we began to understand that there was this systemic inflammatory condition that was associated with SARS-CoV-2, then much more attention focused on using anti-inflammatory and immune suppressing drugs. And the other thing in terms of selecting drugs is there has been a real development in computational methods and protein interaction assays, and therefore it's been possible to repurpose targets from currently approved drugs. But once you've chosen what drug you're going to consider trialing, the next absolute key step is a well-designed clinical trial to provide robust evidence that it works in the condition that you're testing it in. And it's impossible to separate from a well-designed clinical trial infrastructure and collaboration. And therefore, I'm going to take these points all together. So platform trials have really come to the fore in the COVID-19 pandemic. And here are just two trials and the timeline of those trials. One, the recovery trial, which is a UK study, and the other is the Solidarity trial, which is a WHO initiative. And basically, these trials are platform trials in the sense that they trial multiple repurposed drugs within the same trial framework. And I think there are several key things that we can learn from these trials moving forwards. And if we look down the timeline, there are some really astonishing things with the recovery trial. So the protocol for the first trial was written on the 9th of March. The first patient was enrolled into the trial 10 days later. That protocol had undergone ethical, regulatory review and approvals issued within 10 days, something we would never have dreamed of being possible a couple of years ago. And then by the 14th of May, within 2 months, 10,000 patients have been enrolled into this trial. And very, very quickly, by the 5th of June, there was the first readout from the study. So just thinking about what have we learned from these trials and how impressive are they? So the recovery trial has recruited 40,000 participants from 181 sites. And we have now got positive data on dexamethasone and tocilizumab, two drugs. Dexamethasone is now standard of care in the treatment of hospitalized patients with COVID-19 infection. Really importantly, though, it has also given us information about drugs that are not effective. And actually, negative results are really, really important as well. The Solidarity trial was conducted in 30 countries and recruited over 11,000 participants, trialed different medications, and none of those have been shown to be effective. So what can we learn and what should we strive to take forward from these platform trials when thinking about repurposing in other conditions? So the first thing is streamlined ethical and regulatory approvals. The next is readily available funding. Then the network support for research. So within the UK, the NIHR and the Clinical Research Network meant that there were nurses on the ground to support recruiting patients into studies. The next thing is about data collection. When we set up studies, we always want to collect as much information as we can. But actually, in these platform trials, only necessary and then not just the nice to have data was collected. And there was the use of linkage to collect routinely collected data from existing healthcare systems. One of the things that the recovery trial had was a really condensed, concise patient information sheet, a couple of pages long. Many of you will have seen patient information sheets 15, 20 pages long, which is really quite overwhelming. The other key thing from the recovery trial was that trial participation was integrated into routine care. It became standard practice in many, many hospitals across the UK that patients were routinely considered to be enrolled into the recovery trial alongside routine care. There was also early discussion with key stakeholders, namely drug regulators and also the NHS guidance, and collaboration was key. 181 centres across the UK really showed that there was collaboration. But focusing on collaboration a little bit more, and this is some data pulled from the Corona Registry, which is hosted at the University of Pennsylvania. And that has pulled together in one place all the clinical trials that are being conducted on COVID-19. And looking at the top of this slide, there are just 4 drugs that have grade A evidence, strong evidence for a role in COVID-19 infection. I've already mentioned 3 of those: remdesivir, dexamethasone, and tocilizumab. And then also there is good data for baricitinib in treatment of COVID-19. It also indicates the number of positive trials out of completed trials and the number of patients enrolled. And I just want to draw a parallel with a drug, hydroxychloroquine, that was really pushed early in the pandemic, for which there is no strong evidence for the use in COVID-19. And out of 18 completed trials, just 1 had some positive results. However, there are still 258 trials registered as ongoing using hydroxychloroquine. So these are many groups scattered across the globe who are still pursuing this as a potential agent, where maybe if there was better collaboration, we could focus more in those that are showing stronger benefit. So what does vaccines in COVID-19? Does that mean the end of drug repurposing for COVID-19? Is this the miracle solution for COVID-19? Well, vaccines have been an incredibly welcome development in the pandemic, but sadly, not all patients are going to be protected by vaccinations. And there are groups of patients, particularly those that take immune suppressing medications, who vaccines may not protect them against the disease. So moving forward in terms of the COVID-19 landscape within the UK, there's a series of platform trials to test across the spectrum of disease to evaluate repurposed medications. So in terms of the prophylactic spectrum, there's Protect V and Protect CH, 1 pre-exposure, 1 post-exposure. In the community, there is the Principal trial. In hospital, there's the Recovery trial and the REMAP-CAP trial. And then there's another platform, Agile, which tests much earlier phase medications rather than repurposed medications, and then we'll feed them into later platforms. And then also there is Heal COVID, to really recognize one of the complications of COVID-19 infection and the long-term consequences. So just to touch on Protect V, so this is a platform trial that I'm leading, and that's looking at prophylaxis in vulnerable individuals. So these are individuals with kidney disease, on dialysis with a renal transplant, or autoimmune conditions, who may well not mount as good vaccine responses as the general population. And they are currently randomized to either receive a drug called niclosamide, which has been repurposed and reformatted into a nasal formulation, or match placebo. We're at advanced stages of adding in another agent with match placebo, and we have designed so that we can add in further agents to this platform, maximizing efficiency. So what can we learn from this past year, and how can we take it forward? So I think the absolute key is collaboration, utilizing networks, involving all stakeholders. And one of the things I think with COVID-19 is there's been such urgency, we have neglected the involvement of patients. And so patients are absolutely central. We need to streamline processes. Hopefully some of the gains in ethical and regulatory approvals will translate forward into future submissions for other studies. And we need to think about innovative trial designs where we can assess multiple drugs on the same platform, and potentially within multiple patient populations with a similar disease pathogenesis or risk profile. And use these lessons to take forward into drug repurposing in rare disease. Thank you.
Thank you, Rona. That was really interesting and amazing to see both the breadth of research and the coordination across all these different repurposing initiatives in COVID, right? And the potential that could have if you could scale even a tiny fraction of that into rare diseases. It would be amazing what you could achieve. But I was really struck as well by the hydroxychloroquine example, and that was a drug we were all hearing about all the time, right? As something that can make a huge difference into COVID, and seeing the low level of evidence that's out there, but still the persistent pursuit of it is fascinating to see. And maybe just a little cautionary note on what happens when there's such that high degree of urgency and public scrutiny and interest, how people can follow their way down the wrong pathways. So yeah, really, really interesting. Thank you for that. I really enjoyed it. Hopefully Rona's talk has just highlighted the kind of current position for repurposing in COVID and why it's at the top of our attention. And what we want to do now is kind of zoom out a bit and see, is it just COVID that this is happening? Is there really this growth of repurposing beyond COVID? And is it something we're seeing? And hopefully John will be able to dive into that topic for us and walk us through it. So John, please take it away.
Okay, thanks for that. Now I just have to do these small gymnastics and get my slides up. Can everyone see that? Wonderful. Okay. So I'd like to begin by thanking Rick and the whole Find a Cure team for putting together such a wonderful conference. I want to talk to you today about a pervasive idea in drug repurposing, that organizations don't repurpose generic drugs. This is something Rick has already touched on. The reasons organizations are supposedly not interested in repurposing drugs is intimately linked to the law, and I'll explain that, albeit quite briefly. But the primary thing I want to draw your attention to is an empirical study that I've conducted showing that organizations don't stop repurposing drugs. And these results have deep implications for those of us that care about repurposing. I warn you now too that I'm going to ask for a little bit of audience participation halfway through the talk. I won't ask for people to raise their hands. That would be rather foolish. But if you have a pen and paper ready, it might be useful. Okay, so the plan, I'll run you through some background. I'll talk a little bit about patents and what's being called the generic repurposing problem. I'll then talk about how we test whether repurposing stops when generics enter and the results from that test. And then I'll look at some policy implications. Okay, so jumping straight into the background. Why do we care about repurposing? Rick has covered this better than I could, so I'll just leave it to say it's cheaper, faster, and safer than drug development from scratch. What I will say a little bit more about is what is a generic. So in short, it's a drug with the same qualitative and quantitative composition as a reference medical product. That is, it's effectively emulating or copying a product already on market. Generics are relatively easy to produce because they don't have to prove the effectiveness of the drug in phase 3 clinical trials. They just have to prove the qualitative and quantitative composition in bioavailability studies. This makes generics much cheaper than the reference medical product that they emulate. So now I'll say a little bit more about the generic repurposing problem, although I can move a little quicker here thanks to what Rick has said. So I'm going to run you through an extract from Breckenridge and Jacob in Nature Biotechnology in 2019. I should say that Breckenridge is Alistair Breckenridge, the former chair for the MHRA. For those foreigners listening in, that's the equivalent of the US FDA, and Sir Robin Jacob, the former Court of Appeal judge here in the UK, expert on intellectual property and currently a professor at UCL. I'll read through you the extract because they chose their words quite carefully. They said that legal systems of many countries do provide exclusivity regimes that aim to encourage businesses to invest resources to research and develop further uses of existing drugs. You might say, so far, so good. They continued on, though, saying that the problem is that these regimes are not fit for purpose in many respects and do not provide a framework through which drug developers can recoup their investment, particularly for drugs in which generic versions are already available. And then to make things even more clear, they said, if a generic version of a drug is available, developers have little or no opportunity to recoup their investment in the development of that drug. In short, they're saying that if you do develop a new use for a generic drug, you won't make your money back. Okay, so now I'll give you a little more detail on this generic repurposing problem, the legal aspects of it. So generally, the prospect of selling a drug in a competitive marketplace without intellectual property protection, particularly patents, is regarded as an insufficient incentive because competitors might copy the innovator's product and then undercut them in price, having incurred fewer development costs. As we see with generics, this is a phenomenon commonly known as free riding. So the pharmaceutical industry is generally seen as the classic example requiring patent protection. And for new drug compounds, this is generally thought to work quite well. where the organization can patent the compound, stopping all commercial uses of that molecule for 20 years. But the situation is different for repurposing. Because the compound has already been claimed, the patent only claims new uses of that drug, which are much more narrow. And these claims are actually found invalid much more frequently than patents on new compounds. Sorry, I should have clicked through my slides. And then even if that patent is valid, it's difficult to enforce due to a phenomenon known as cross-label use, where a generic version of that drug that hasn't actually been specifically authorized for the new use is still dispensed for the repurposed indication. This happens due to the way drugs are prescribed, dispensed, and reimbursed. And an example here is pregabalin, which was originally patented and used to treat epilepsy. The compound was then found effective and patented for the treatment of pain. But this patent for pain was found invalid in the UK. And then even if the patent was valid, a court said that when generic pregabalin was sold saying it only treated epilepsy, but it was dispensed for the use in pain, the generic company that produced the drug was not liable for this use. So there in a nutshell is the generic repurposing problem. And now what I want to explore is whether that is true. So the impact of generics on repurposing was first described in 1999 by Pulcinelli, and it's probably known for much later than that. Sorry, much earlier than that. But if Breckenridge and Jacob accurately state that developers have little or no opportunity to recoup their investments, then what this should mean then is that developers should effectively stop conducting clinical trials to repurpose drugs once or immediately before a generic enters the market. And I'd just like to highlight here, this is what I'd like to test. By looking at clinical trials, can we see that drug developers stop repurposing drugs? So let's see if that's true. How do we do that? So the first step in this was to identify all drugs, or 16 drugs that went generic for the first time in 2014. And I'm focused here on the US because it's the most valuable medical territory in the world. So therefore I used FDA data. I then downloaded a list of all adult phase 2 and phase 3 clinical trials begun for these drugs between the authorization date and 31 March 2020, when I downloaded this data from TrialTrove, which is a commercial level database of clinical trial information. I should mention though that this was not limited to rare diseases, or the clinical trials were not limited to rare diseases. The second step in the method was then to analyze these clinical trials, over 2,000 clinical trials. And I classified each clinical trial according to two criteria. One was who, where I classified it as originators, so those that originally produced the drug, competitors or commercial competitors to the originators, and then the public sector, where we can look at things like governments, universities, and hospitals. The second criterion that we looked at was the type of clinical trial. And here classified it as either the same condition, so it's looking at the efficacy of that drug, perhaps in or biomarkers, new pharmaceutical forms, or perhaps new patient cohorts suffering the same condition. Or the alternative was to label the clinical trial as a new condition, and that's a new or different medical condition compared to the preceding condition that the drug has already been authorized. And we did that with reference to the International Classification Diseases version 11, which is a database maintained by the World Health Organization. And then just to emphasize here that roughly half and half of the trials that we looked at, half of the 2,000 were for new condition. And for the rest of this talk, I will concentrate only on new condition clinical trials, or primarily talk about new condition clinical trials, because those are the ones that we're really interested in. So jumping straight through to the results. What we're looking at here is the originator trials for all 16 drugs. On the y-axis, we're looking at the number of trials. You should be able to see my cursor. And then on the x, we're actually looking at the years leading up to the generic entry in year 0. Year 0 roughly equates to 2014. All these drugs went generic in 2014. But because the generic entry date is different for each of the 16 drugs, I've had to use this sort of -1 to +5, which is 5 years after the generic is entered. But it roughly equates to 2014, and then I've used 5-year increments there. to illustrate this. Now, as you can see, I've blocked out 6 years leading up to generic entry and then the 5 years after. And this is the crowd participation part. I want you to predict what happened in the lead up to the generic entry and then afterwards. So maybe just think, has the number of clinical trials decreased? Has it remained steady, or has it increased? And here we're just talking about the originators, so the companies that originally produced the drugs. And I'll give you a moment just to think about that. Okay, well, here's the result. It decreased, and quite markedly, probably consistent with Breckenridge and Jacobs' prediction. But here we're looking at the same data, including originators and competitors, so originators in blue and competitors in orange. And as we can see there, that actually when the generic enters, we see a fairly marked increase in competitor action. So these are commercial competitors to the originators. So trials there don't stop at the commercial level. But now I'd like to show you the trial results from the point of view of the public sector. So this is the same X and Y axes as we saw for the originator trials, but now I'd like you to predict whether you think the trials from 6 years before generic entry and then after, do they decrease, increase, or remain steady? And just to illustrate, I've separated it between government and university data, but for these purposes, just think of those two as a whole, public sector trials. So I'll give you a moment to think about that too. Okay, perhaps for those who looked at the titles for my talk, you've realized that it's actually generally increasing, and I'm working on the statistics just to show that. I should also say that there's been, the 2019 trials have actually increased as well, but there was a data collection point, so I need to update those numbers of trials. So what we see there is that the trials are actually increasing for the public sector once the generic enters the market and immediately before. Now, this has taken me by surprise and many people that I show this data. And so I think what we need to do here is immediately think about policy implications from this. So first, thoughts on why public sector trials increase. Rick touched on this. It's cheap, safe-ish, and easy to access. And it's worth emphasizing here that there's another 1,000 trials on these drugs that were for the same condition. So there's lots of clinical information on these. I think it's also worthwhile thinking about here that one of the reasons the trials here are so high for the public sector is that there's thousands of universities and hospitals around the world, and it's much higher compared to the number of pharmaceutical organizations. It's also worthwhile thinking about why do it. Some of the hospitals and academics would have been thinking about the commercial benefit, that is linking up with a generic or a competitor or the originator, and then eventually changing on-label use. But there would have been a modest amount of that. We also know that a lot of this is targeted towards off-label use, which has already been spoken about, but is generally suboptimal to actually changing the on-label use of these drugs. Many people would have also done it for the first to publish and career progression route, something we're very familiar with in academia and what is often called open science. Of course, there's a huge component of altruism here too. Scientists and clinicians want to treat the patients in front of them. So I think in these last few slides, I'd like to think about how do we capitalize on these public sector trials? Vast numbers of public sector trials are being conducted. So interestingly, several attempts are being made to capitalize on them. There's the ILP in the UK, the Innovative Licensing and Access Pathway, that began in January this year. One of its arms is to focus on academic repurposing, amongst other things. Then there's the STAMP trial, the Safe and Timely Access to Medical Products, which is being run by the various organizations in the EU and headed by the EMA. And what they're looking to do is the idea of recruiting academic champions that the EMA supports with advice and perhaps some funding for research. And then they're hoping to partner them with either originators or generic companies. There's also a similar thing operating in the US with NCATs. But it's fair to describe all of these are very much in a learning phase, which we don't know which of these is working well, and they're all taking quite different approaches. So I think this is a topic that really calls for bold policy action. And in this respect, the UK or several members of parliament attempted with the off-patents drug bill that would have allowed the government to seek licenses for off-patent drugs when there was evidence they could have clinical effect. But this created a problem in that the government would have been seeking licenses as well as operating as a regulator, a conflict of interests. So that bill was never passed. There's lots of possibilities, and I think there's lots of ideas here, and I'd really like to hear from people to hear what they think. And of course, there's Find a Cure's social impact bond, which I think could play a large role here. And finally, there's potential for incredible impact here, because in another study that I'm writing, we can show that only actually 22% of EU orphan drugs come from a repurposed background. And I'll finish there. Thank you.
John, thank you so much. It's great to hear someone else talking about some of these things I've been banging on about for a few years, and actually some empirical evidence to support this concept that repurposing is happening in the labs, in the hospitals from an academic perspective. And I believe a lot in the rare disease space, and that we need to think about it and try and think about how that impacts the policy. So guys out there listening, we'd love to hear some of your thoughts and questions around this topic and how we can drive this forward. So please do submit them for our Q&A coming up shortly. But as perfectly planned by the team here, this really nicely segues into our next speaker and his perspective on repurposing. So I'm really pleased to introduce Indy Banerjee. So Indy's going to be kind of giving his insight into actually being one of these academic clinicians, essentially at the coalface, treating patients and thinking about how you can deliver impact to those patients on a daily basis. So Indy, really happy to have you, and please do take it away.
Thanks, Rick. If I could just, thanks for putting my slides up. So I'm just going to take a little bit of a step back to see, connect to the patients, because that's what it really matters. My experience comes from the rare disease of congenital hyperinsulinism, which I hope I can just give you a little bit of a quick short summary. If I can try and move the slide on. So this is Jack, who was, this picture is from the Daily Mail. You can see he is quite large, and this is obviously, you can read the calendar date, which is 2007. So he is 14 years old. So when Jack was born in a hospital in Manchester, he was quite large. And for those of you who know what babies' birth weights are usually, he was quite large. In fact, he was one of the largest in the country for his gestational age. And he started developing hypoglycemia from day one. His mother had developed gestational diabetes, so the tendency was to say, oh, this is just an infant of a diabetic mother, which often is a common scenario. Children get hypoglycemic and this condition resolves. Of course, Jack didn't resolve. He needed increasing amounts of dextrose, and you can see from the lines going into him that he was needing high concentration dextrose. We tried medication that was there at the time, which is diazoxide, which acts on the potassium channels and hopefully stabilizes excess insulin production, which is what's happening here. But he didn't respond to it. He also did not respond to another medication to reduce insulin secretion. So this is octreotide, which is a somatostatin receptor agonist, and you can see that we ran out of medication, and the only choice in front of us was to do a subtotal pancreatectomy. So the idea is that you reduce the insulin load from the pancreas and you hopefully achieve cure. So we did achieve cure in that he was no longer hypoglycemic. But at the age of 13 years, so last year, he developed diabetes. The important point, though, is that he is well. He goes to a grammar school and he's doing really well. And the reason to say this is that hypoglycemia is a really high impact condition that affects your brain. And in Jack's case, although it needed quite a bit of medication, he didn't have this, which is quite a lot of brain damage. What you see here on this slide, PVHSI stands for periventricular high signal intensity. Just tells you that with hypoglycemia, you have infarctions or ischemia to parts of the brain. What's really telling is the back of the brain. The OLA stands for occipital lobe atrophy. Now, the occipital lobe coordinates what we see and what we understand of the vision that we see. So this poor child who has this brain injury is able to see. His eyes are fine, his optic nerves are fine, but he does not understand what he sees. So effectively, he is visually cortically blind, which is quite sad, because this particular child that I have put up there, MR scan, has simply a what we call transient. So the short-term hypoglycemia ended up with such catastrophic damage. So what I'm trying to tell you is that, look, although we did a subtotal pancreatectomy, Jack did not end up here, but a number of children. In fact, in our experience, 30% of children end up with brain damage. Worldwide, about 50% of children have some kind of neurodevelopmental delay and some kind of neurological disorder. This will carry on. Neurodisability is lifelong. So it's something that is quite worrying. If I could move on to the next slide. So you can see that there's a pressure on us to innovate, repurpose, use different types of treatment that are out there. And this seemed to be like the perfect treatment. So a group, my colleagues from London, they put together a paper. You can see this is a very high impact journal, New England Journal of Medicine, and they did an observational study using this drug called sirolimus, which is used also as an immunosuppressant and is used in children having transplantation. It's also used in adults. So most of the data is from adults, less so for children, and certainly not for very young children. So Jack is a little baby. For Jack's age, this has not really been tried and tested. However, this is the pressure of having few medicines, high impact condition. I need to make it better. What can we do? And therefore, we move towards using sirolimus. So initial reports from this group are very effective. When we tried using it, this is in a collaboration, and this is key. It comes back to the points that Rona and Rick, you're making, that we need collaboration for rare disease, repurposed medications. And so we tried to form a collaboration and did what we thought was an observational study rather than a unified protocol-driven study. Our effects were not as good, and what we really saw was very significant infections, so much so that these children started developing fungal infections. And as you know, fungal infections, once they're set in, is really difficult to get rid of. What was also striking is a few children started developing exocrine pancreatic insufficiency. Now, that happens when you take somebody's pancreas out, you lose the endocrine bits and you lose the exocrine bits. In this child, or these children, you haven't taken out their pancreases, but you're starting to see the effects of real pancreatic damage happening with medication. And that's as bad, even worse than pancreatectomy. So our experience was not so good. Now, we talked about the pathways of disease, and it is really important to understand the pathway of disease better. The pathway postulated for this drug is the mTOR pathway, which is post-insulin receptor signaling. And there is the prospect that it reduces proliferation pathways downstream of insulin receptors. So if I could have the next slide, that hopefully will show me that when we actually took the study into the lab and looked at islets and whole pancreas proliferation using standard markers of proliferation, what you see here is the black bar and the gray bar. There is no real difference. So the proliferation did not settle to control levels, which implies that the proliferation pathway, that obviously is an important part in older children, adults, is not really playing a part in the pancreas in these children with congenital hyperinsulin's. So again, it comes back to the point that repurposed medicine in a different condition may be different when we apply to a completely different population, a completely different disease. If I can move on to the next one. So my lessons, I suppose, is I do need to understand that the drug action and mechanism, and yes, it may be well established in that particular case, but I need to establish in my scenario. If you're going to repurpose, I would think that you should have effects and side effects being analyzed carefully. And the effects you can only work out by trying the drug or repurposing in a form of a clinical trial, but the side effects have also to be carefully evaluated. In this particular case with sirolimus, we were really at a loss and struggling to keep these poor children alive. And thankfully, we were okay, but we certainly learned our lessons. We built up a protocol within our center, and we shared the protocol with France, the French group in this particular study. And although they were not exactly the same, they were very similar, which really helped. But if you're going to repurpose, again, comes back to, I think, what Rona was saying, we need to capture this in the form of a clinical trial, gather the data, because that's really going to be valuable. Most importantly, I would suggest that we do this as part of a funded study with all the rigorous checks and balances, and it could be observation, it could be case control. However, it needs to be set up like a clinical trial, and just trying it sounds rather maverick and cavalier. It's not particularly good for patient safety. And be open-minded about outcomes. Great if it works. If it doesn't work, that's also negative results. Again, coming back to what John, Rick, and Rona were saying, you have to be open-minded about outcomes. So yeah, so I think that's what I have to say. Is it's a bit of a salutary lesson. My lessons are that you can try, but try it within the parameters of what we think is a convincing case. Do it within a trial configuration and share and collaborate. So thank you.
Thanks, Indy. It's really, really helpful and really interesting to see that kind of challenges you face, the lessons you learned, and also just to reflect, I suppose, on a moment on that position you're in, really, seeing these patients day in, day out, the situation being that the only thing you can do ultimately for some patients is an extreme operation to remove a pancreas, and that impetus that is clearly present for people in that situation to try and find a way to do things. And I think fundamentally what is needed is some more support, whether that's in funding, obviously primarily, but training concepts and collaborations to help move things forward in a properly researched and evidenced way. And that's amazing, the work you did to try and move that forward, and amazing also the way you identified the challenges there and just made the decision to not carry on. Have you got any kind of personal reflections on that side of it?
So we're working on different types of clinical trials, and I have to admit that when you try and repurpose in a clinical trial setting, and I come back to John's point that a lot of these drugs are now generics, and therefore you should be using them in clinical trial formats. However, if you went to funding agencies within the UK, most of them, I have to admit, are more geared towards big diseases, so they're looking for bigger value. And this is why I think the rare disease community needs to make some noises and make deputations that, look, that is fine. We certainly want dementia, cardiovascular cancer. We do not want to ignore rare diseases, and there should be a pathway, which unfortunately there isn't at the moment. NIHR, so National Institute of Health Research, which is the research arm of the NHS, I think have started to wake up, and they're saying, right, we will help you. You develop your research ideas, we will help you. And what we're doing at the moment within endocrinology, outside congenital hyperinsulinism, is bring people together from different rare endocrine disorder patient communities and ask them, right, what do you think are your top 3 priorities? Let's work them out into a small A4, what we call the PICO model, and then work with NIHR to bring them forward. So we are trying our best in every possible angle, but at the moment, the opportunities are not brilliant. I'd be keen to hear from others what they think.
Thank you, Indy. And hopefully we're going to have a decent amount of discussion on some of these very topics during the workshop this afternoon. And I'd love to hear the opinions of our audience on this and how we can move this side of repurposing forward effectively. So now we're just going to go to our final kind of talk piece to complete the narrative, really, which is returning to Rona and looking at a situation where actually one of these research studies did get some funding and was able to generate the evidence in that kind of regulated clinical setting and move things forward for patients. So Rona, I'm going to try and share my screen to give you the slides, and please do hopefully dive into this final part of our story.
Great, thanks very much.
Can everyone see those okay?
Yeah, so if you move on to the next slide. So as I said, prior to COVID, my work really focused on a condition called vasculitis, which basically is inflammation of the blood vessels. You've got blood vessels everywhere in your body, so it can affect any organ in your body. And it's classified by the size of the vessel that it's affected, large, medium, or small. And one of the small vessel vasculitides is called ANCA-associated vasculitis, and it is a rare disease affecting about 1 in 10,000 people. Next slide. Next slide. So, go back. So as I said, vasculitis affects every organ. So classically, it can affect the nose, destroying the cartilage. You get a saddle nose deformity. You can get masses behind the eyes. You can get masses within your lungs. You can bleed into the lungs. And in the histology, it can affect your kidneys, and you can get rashes on the skin. And actually, it can be organ and life-threatening. Next slide. So there was no treatment at all until about the 1970s, when steroids, prednisolone, and cyclophosphamide were used to treat the disease. And really, nothing changed until early 2001. And basically, what happened, a patient presented, they were given steroids, cyclophosphamide, an immune suppressing drug, and then a different drug, azathioprine. Next slide. And invariably, in the majority of patients, they would have a relapse of their disease at some point, and they'd need more steroids and more cyclophosphamide, and they'd have another relapse. And every time they had a relapse, they were exposed to more of these drugs that had toxic side effects, and they accumulated damage to their kidneys and their lungs. Next slide. So as I said, back in 2001, there was a drug around called rituximab, which is an anti-CD20 monoclonal antibody. CD20 is a receptor that's expressed on B cells. And so if you give a drug, rituximab, it just depletes the B cells. And it had its original license for B-cell lymphoma and rheumatoid arthritis. Next slide. Now, just focusing in the red circle here, as I said, ANCA vasculitis is dependent on this antibody called ANCA. And actually, the ANCA is produced by the B cells. So the theory was, if you deplete the B cells, you didn't generate the ANCA antibodies, and therefore you didn't trigger this disease process. Now, this is, of course, a very simplified schema of the disease pathogenesis, but that was the rationale for taking out B cells. And it was used, as we said, off-label in the most severe cases where everything else had been tried. And there were case reports and case series of successes. Next slide. So then there were 2 pivotal trials that really made a game change in ANCA vasculitis. And as I said, traditionally treatment was steroids, cyclophosphamide, and azathioprine. None of these were licensed drugs for vasculitis. There was no licensed drug. But these 2 studies that were published, one was by the European Vasculitis Society, a collaborative network, and the other was by the Vasculitis Clinical Research Consortium, a US network. And they basically conducted trials comparing rituximab to cyclophosphamide in ANCA vasculitis. Next slide. Now, I won't go through in detail the results, but in essence, both were positive trials. showing that rituximab was not inferior to cyclophosphamide. And in the RAVE trial, they showed that actually in a subgroup, it probably was superior to rituximab. So it was better than cyclophosphamide in those with relapsing disease. Next slide. And the FDA acted very quickly on this. And in 2011, it issued approval for rituximab for induction of remission in ANCA-associated vasculitis. And very quickly within the US, it has become standard of care to use rituximab rather than cyclophosphamide as first-line treatment. Next slide. However, in the NHS, despite approvals, there's almost another layer of, even though drugs have been shown to have efficacy, they need to be shown to be cost effective. And so NHS England in 2015 offered guidance of when rituximab should be used in patients with ANCA-vasculitis. And basically they needed to have failed cyclophosphamide, or that cyclophosphamide wasn't tolerated or contraindicated. So for example, cyclophosphamide is known to impact fertility, and therefore in women of childbearing age, rituximab was the preference. But after these trials, the induction trials, there was another problem. One, it was the relapses. So next slide. So then there were two studies that had been done. Mainritsen was done by the French vasculitis study group, and ritazaram was a study that I led, and that was a collaboration between the Europeans and the Americans, and it also ran in Australia, various countries in Europe and Japan. And that was a combination of charity-funded, government-funded, and industry-supported study. And both of these trials showed that repeated doses of rituximab was better than azathioprine at maintaining remission. Next slide. And as before, the FDA acted very quickly, and they extended the license for rituximab to include remission maintenance for ANCA-associated vasculitis in October 2018. Next slide. Now, as with NHS England, not quite so straightforward, but rituximab is allowed to be used as a maintenance agent if a patient is either in a randomized trial, they've already had rituximab and have had a relapse, or cyclophosphamide is contraindicated. But in order to collect data, this needs to be done within a specialized center, and we need to register them in the UKVAS database and also consider future trials. So next slide. So rituximab is the first licensed treatment in ANCA-associated vasculitis. And this has kind of taken you through a story of a clear unmet need in the disease. There was a sound scientific rationale for using rituximab initially as off-label, but then developing the studies. These were academically initiated trials, but would not have been possible without industry collaboration. And there are various combined funding models, including charity funding, government funding, pharma funding, and it utilized existing networks. So the UVAS, the European Vasculitis Society, the VCRC, and the French Vasculitis Study Group. So hopefully that's a quick whistle-stop tour of 20 years of rituximab in ANCA-associated vasculitis, but it has been a real game changer for this condition.
Thank you so much, Rona. Let me clear that. And I think hopefully what we've all done here is taking you through this crazy whistle-stop tour, firstly looking at the basics of repurposing, beginning to try and convince you of its impact, and then looking at how it's actually relevant now, why COVID has driven it through, and the fact that actually there's a huge amount of repurposing going on behind the scenes. And it's in many cases the academics, the researchers, the clinicians that are driving this repurposing, particularly in our areas, to try and meet the unmet need that we're seeing out there. And thank you all for your contribution to illustrate that. Now, what we're going to do is try and dive into some of the questions that are being fed through the chat and that some of the audience are speaking about. So thank you all for engaging with this so far. And one of the early ones that came through was tied into the route to market for studies that are clinician led. And I just wondered what you guys think and see as the kind of the route for these academic-led trials to actually reach the patients ultimately? What are the major routes you're seeing, and what do you think the ideal route is? So I don't know, Indy, I just saw you on mute a minute ago. I don't know if you've got any thoughts on this to start with.
So I thought that the route depends on the type of disease and how long the study takes and back into the patient. So, you know, there is a convoluted route, but I think with orphan drugs, there is the willingness to speed things up. And certainly for drugs that are being trialed, maybe not repurposed, I think both MHRA in the UK and FDA in the States, they accept that it's not quite the same as, say, diabetes, and they don't need thousands and thousands of patients. They can do with smaller number of patients as long as a well-done, well-conducted study. It still takes, on average, in a study setup to actually doing the study. Now, quite often in children, what happens is these regulatory agencies will ask for the study to be done in older children and then come down to younger children. So again, that takes a while. So it still takes, 3, 4, 5 years easily. And that's something that we are conscious about, and painfully so. But it's unfortunately is the amount of time. You can talk about what are the incentives for these biotechs or industry that take up these rare disease trials and drugs, because you could argue that they're not really going to make money, and I'd be quite keen to hear from industry what they think. But it looks like that there is a certain amount of confidence and interest in biotech to develop orphan drugs because it's faster, the returns are greater, and this period of where you hold the patent is probably a little bit longer. So that there are positives, but for repurposed drugs, I still think that if you run this within the remit of a clinical trial, it's still going to take 2, 3, 4 years, depending on the drug and the condition.
Cool, thanks, Andy. Rona, any thoughts on this topic, particularly pulling on that sense of collaboration you just highlighted in the last talk there, and that seems to be really key to drive this to success and actually get the license and access to patients in that case?
Yeah, so I think in all of this, collaboration is absolutely key, and it's collaboration amongst clinicians across specialties, but more key is collaboration across industry, with patients, with regulators, and I think that is the real lessons to be learned from this year, and the driving force forwards.
Cool, thanks, Rona. John, any thoughts on this? I mean, what are you seeing some of the patent work you're doing? Is there a prevalence of these projects falling through, or is it really moving into that off-label space very, very often from the early applications?
From what I gather with a lot of that data that I looked at, it is predominantly off-label. And we see increasing number of cases being litigated about off-label use as well, which I think is a little unfortunate. And I think, there's something to learn here from what Rona was talking about with COVID, is that we see, incredible collaboration. We see early stage interaction with the regulators, and the regulators with dexamethasone and things like that, like the EMA, they changed those labels without a marketing authorization holder actually submitting an application. That's incredibly unusual. But, if we want to think about using the data that a lot of clinicians are producing, then we have to start thinking about what we can learn from COVID and how we can break down some of these barriers to get them across. And maybe it is getting that scientific advice from the EMA about how to design those trials so they'd be accepted for changing labels earlier and things like that.
Thanks, John. Kind of feeding off that then, what do we think that academic research clinical community needs to try and help move some of these repurposing projects, particularly those in the generic space, forward more effectively? I mean, do you have general opinions on that and things that we would like to see in place to help this, particularly in rare diseases?
So Rick, if I might start, so we had a bit of a process looking at different companies and if they had any compound libraries that they would happily share with us. So big companies, big pharma often produce lots of compounds, but they don't always use, and whether they'd be open and transparent to collaborate. Again, the buzzword collaboration. With interest partners, and they have done this in the past, but not really been very successful. I just wonder whether we could boost this in any kind of way with bigger incentives, maybe even incentives from MRC, from government in some shape or form, that you end up with, you encourage these repurposed generics to bring to the market. And if there are drugs that are in compound libraries, maybe not repurposed, if they've been used for something else and didn't work or didn't try, then why not share, and so we can do healthcare better in this rare disease space. So that's one angle. I thought we as clinicians needed to be a little bit more educated. What is the model for repurposing? And I think we're just finding our way. People who are in this space, Rona obviously did a lot of work, but most of us at the coalface don't know exactly how to go about our way of how can we bring a drug to trial or kind of study.
Thanks, Indy. I think it's providing that support into people in the situation that you're in to give you guidance and thinking about how to move through that area, but also the collaboration that we've already talked about, and encouraging those networks to come together to find ways to make most success about it. Rona, anything to add on that point?
So I think just maybe taking one step back from being a clinician, and it's actually going to the patient. So often as clinicians, for many conditions, we think we know what's important, but actually what we think is important may not be important to the patients. And so I think patients need to become central to this process so that we're asking the right questions of repurposing and we're measuring the right outcomes for repurposing. Some conditions, it may be obvious that, for example, there is a life-threatening outcome. But very, very many conditions, it is the symptoms that are debilitating for patients that clinicians shy away from because they're difficult to measure and difficult to prove outcomes for.
Thanks, Rona. That's really nicely segued into some of the questions we're getting in from the chat, actually. And there's someone who's talking about their work in GLUT1 deficiency disease, and was just really, in summary, asking how patients and patient group organizations can begin to help move things forward in the drug repurposing space or the clinical development space, and what your opinions might be as to where patients can feed in. Rona, I know you did some work in this area, but I don't know if anyone else has any thoughts as well.
So Rick, I was just going to add that we've asked, so in congenital hyperinsulinism, we have regular meetings with the charities. So the UK-based charity is UKCHI. You also work closely with the international charity, and they're also driving, it's in their interest to drive research. So it works, and obviously it's not happened and borne fruit right away, but if we persist with that collaboration, that will happen. We're taking it into the British Society of Pediatric Endocrinology and expanding to other rare diseases, and again asking the same question that Rona asked, which is what is important for you guys, not from the doctors, nurses perspective, but from your patient family perspective. So we are starting to see a massive movement. I think it'll be even better as we provide those pathways to different groups.
Thanks, Indy. Anything further to add on that point, Rona? Because I know you're heavily involved in the kind of patient side of research.
Yeah, so we've developed here in Cambridge something called the Patient-Led Research Hub, which is just a small venture at the moment, but it enables patient charities to bring ideas to the table of research questions that are important for their patient populations, and then to discuss whether it's feasible to conduct a project or study and whether that can be pushed forward to kind of a grant application stage, and we partner up with relevant academics in the field. So it's not as big as we like. We're limited by resource, but I do think that it is a step in the right direction.
Thanks, Verona. John, were you going to say something?
Just one thing to add there. I know the European Medicines Agency recently made scientific advice free to SMEs, things like that. Normally, this scientific advice costs 60,000 euros, and when you're starting to plan things like clinical trials, for what they would actually accept, because I think one of the things that Indy hinted at before is that for lots of clinicians, the idea of repurposing and designing these things can be relatively new, and that's a heavy burden to carry because, of course, you guys are trained in keeping people alive primarily. And so to design clinical trials on top of that is a lot to ask, but there are a lot of these things available and they're increasingly coming online, and these types of resources should be taken advantage of as much as possible.
Absolutely. Thanks, John. Just another one probably to you, John, actually. A question's just come in asking about the situation around pricing within the NHS and whether or not a license price can be set for different conditions, or whether you can have a single price for all. And I think this feeds into that issue around patents and off-label prescription. I don't know if can you comment on that, or would you be able to?
Yeah, it's a little bit outside of my wheelhouse. Pricing is a complicated one, and it feeds into that issue with cross-label use that I spoke about during my talk. Because if you have the same drug that is authorized for different indications, but you're charging different amounts, then generally within the NHS, they try and use the cheapest source. And so differential pricing may not work too well.
Yeah, and go on.
Yeah, in other countries, it does work quite well. Denmark works that way, but generally not.
Yeah, and in the NHS, my experience has always been that multi-indication pricing is something they really move away from and don't want to engage with and talk about. So, if there's a drug license for a condition, if you then license for a different condition, it will retain that same price, and you can't vary the price between those indications, which has some implications from how you can go about repurposing and create that business model.
Yeah, but as you spoke about, like that reformulation, that change of dosage, that change of route, all of a sudden you actually have a different medical product.
Exactly.
And look at different medical pricing.
Yeah, completely.
Rick,
yes,
I was just going to add that NHS England in the UK, they commission highly specialized services for rare diseases. So if they have a contract with certain providers, they may choose to have a differential pricing, which nobody really knows. It's fairly opaque. So it's a smoke and mirrors for a lot of us, really.
That's really good. Thank you. Getting towards the close now, one of the questions I just wanted to touch on, which was asked, obviously we talked about the role that patients can play in helping support design trials, but obviously patients need to attend and be part of trials themselves, and there can be a very high burden for that. And someone's just asking if you know of any central fund or kind of process in place to help support patients to attend clinical trials or take part in them, or is that just something that's worked on a trial by trial basis? I don't know if Rona or Indy, you have any thoughts on how that's delivered in your experience?
So as far as I'm aware, there is no central fund, and often there may be a patient travel cost as part of a trial. But it is a big issue. However, again, I hope it might be one of those things that we benefit from COVID. So a lot of trial work has moved to be remote, where there are some remote follow-ups, and potentially, if that translates through, there'll be less study visits. And I think the other thing that we could really utilize is routinely collected healthcare data as well. So link with existing data that is collected through NHS Digital, so hospital admissions and links with GP data. So it can be a barrier for some, and we recognise that, but I don't think there is a central solution, I'm afraid.
Thank you, Rona. So I'm just going to give you guys my closing question now before we get ready to think about the rest of the conference. And I'd just be interested if each of you could maybe have a little thought on what you think is needed to really help drive success in rare disease repurposing in the future. Indy, if you'd like to dive in to start with.
Yeah, so my thoughts are collaboration between different partners, so clinicians, patient groups, and regulatory agencies. So how can we bring existing or non-existing drugs to the market? But also, I would welcome greater responsibility from the funding agencies, maybe charitable, maybe government funded, so that we know that there is it's not a black hole that we are pitching at, that we know that there is a way to get funding which would patient benefit, but it has to be a collaboration driven approach.
Thank you, Indy. John, I'll pass to you.
Yeah, I think it's has become really apparent with Indy and Rona here in front of us that a lot of repurposing this space is clinician driven, and that's probably one of the centers that's, you know, the real wheelhouse for these things. And once we realize that a lot of things orientate around that, then it's how to get funding and support for people like that. And I think we can start looking at tweaking the system, particularly with some of the insights that we've gained from coronavirus and that Rona outlined.
Thank you, John. And Rona?
And so I think probably what we need to do is become a little bit more innovative in our trial design. So can we think about several candidates for one condition that could be tested in the same platform? Or should we even think about several different diseases, maybe use the same pathogenic pathway or a particular target, and could they be considered in the same platform? It is not straightforward. There are very complicated design aspects, but I think unless we think, we will stifle the possibilities that we could have for drug repurposing.
Thanks, Rona. That's a really nice kind of note and message to end on. And with about a minute to go, I'd like to thank all three of you for huge contributions to this session. I've enjoyed it hugely. I think it's gone really well, and I think we've really helped to kind of sew the story together that we're hoping to, and to see the questions that have been coming in and the reaction on Swapcard in the chat has been really positive. So thank you everyone for listening, and thank you three for your contribution. Before we depart, I am duty bound just to do a few little plugs and housekeeping. Firstly, I just wanted to highlight and thank again the event sponsors who have been really helping to drive this event forward and make it possible for us to be here today. And secondly, I'd like to shout out to the audience. We've got a huge amount of things going on today, and there's a lot of chances for you to interact. So coming up in 15 minutes or so, you've got the chance to join the pitch and mix sessions and network and hear some short stories from delegates and really talk to each other and share your experiences. And later this afternoon, really excited to have our kicking off kickstarting drug repurposing sessions hosted both by LifeArc and Helix, where it's really a chance for you to get your teeth into some of these topics, dive into it and discuss with each other and help to come up with new ways to move this field forward. So please attend that. Thank you all so much for listening. Thank you to my speakers, and we're looking forward to you all joining us for the rest of the event. Thank you and goodbye.
Thanks.